Good afternoon, everyone, and welcome to our webinar titled Rediscovering Decode Duchenne. My name is Justin Layton, and I'm the Director of Genomic Marketing and Product Management at PerkinElmer Genomics. I will be both a panelist and the moderator for this afternoon's joint presentation from Parent Project Muscular Dystrophy and PerkinElmer Genomics. The goal of today's webinar is to introduce everyone to Decode Duchenne, a no-cost genetic testing program offered by our laboratory in PPMD, along with our industry partners Sarepta and PTC Therapeutics. Today's presentation will include a discussion about the history of the program, who qualifies for testing, and the technologies used for diagnostic testing. We'll also review recent updates to the program that have been implemented to increase access to and ease of use for the program. Today's webinar is scheduled for approximately 30 minutes, which will hopefully leave time for a short question and answer session at the end. You may submit your questions at any time during the webinar through the chat or question box. Without further ado, it is my pleasure to introduce our additional speakers for today's webinar, Ann Martin and Dr. Madhuri Hegde. Ann Martin is a certified genetic counselor at PPMD and currently serves as the director of the Duchenne Registry, as well as the Decode Duchenne program. Dr. Hegde is the vice president and chief scientific officer of Global Laboratory Services at PerkinElmer, where she's responsible for 6 clinical laboratories around the world. She's a board-certified molecular geneticist who has been studying neuromuscular disorders for over 20 years. Today, Ann will start with an overview of the Decode Duchenne program, while Dr. Hegde will subsequently dive into the testing technologies employed throughout the program. Ann, the floor is yours.
Great, thank you, Justin. Next slide. All right, thank you all for joining us today. I am thrilled to talk with you about Decode Duchenne, which is a program of Parent Project Muscular Dystrophy. PPMD is the largest nonprofit organization in the United States focused solely on Duchenne and Becker muscular dystrophy. Decode Duchenne was actually launched back in 2013 in order to provide free genetic testing and counseling to patients and families affected by Duchenne or Becker muscular dystrophy. We really recognized the need to lower the barrier to genetic testing for the Duchenne and Becker community, to accelerate diagnosis, and to shorten the long diagnostic odyssey that many families face, but also to increase access to new therapies being developed for Duchenne and Becker. Next slide. As many of you know, genetic testing is critical for Duchenne for several reasons. Of course, it is the gold standard for diagnosis of Duchenne or Becker. It is also essential for family planning and for carrier testing of the mother and other at-risk females in the family. Approximately 2/3 of all boys who are a new diagnosis in a family have a mother who is a carrier. And a positive carrier test can then lead to reproductive options, such as prenatal testing and even pre-implantation genetic testing. And last but not least, genetic testing is crucial for mutation-specific therapies, such as exon skipping and nonsense mutation read-through. There are 2 mutation-specific therapies that are now approved in the US, and there are several more in the pipeline and currently in trial. Next slide. I want to thank our industry sponsors, both Sarepta Therapeutics and PTC Therapeutics, who generously fund the program. Decode is really a great example of a beneficial partnership between an advocacy organization and industry and how they can work together to positively impact the patient community. I do want to emphasize that only de-identified data is shared with the industry sponsors, so no patient names or other identifying information is shared. And both the clinician and patient must provide consent to share the de-identified data. And our industry sponsors use this data to better understand Duchenne and Becker and to better plan for clinical trials. So since we launched Decode Duchenne, we have provided over 6 years of free genetic testing to families. Over 425 clinicians across the US and Canada have used our program. Over 250 women have had free carrier testing since we added carrier testing 2 years ago. and over 1,200 patients have received diagnostic confirmation of Duchenne or Becker. And it's worth noting that 90% of patients tested through our program are found to have a pathogenic variant in the DMD gene. Next slide. So patients are eligible for Decode if they are either a male with a confirmed or suspected diagnosis of Duchenne or Becker, or a female with a relative with Duchenne or Becker with a known mutation. This program is not only for patients who have never had genetic testing, but also for patients who need repeat testing because previous testing did not confirm a mutation or is simply outdated. Any type of financial barrier qualifies a patient, and patients must reside within the US or Canada. Next slide. So for the available testing options, I did want to point out that you can order creatine kinase activity. You now have the ability to order a CK-MM level at the same time as genetic testing when you use either a dried blood spot card or a whole blood sample. I do want to point out that the new whole blood kits for the Decode program now have a sodium heparin green top tube for collection of the CK-MM, as well as a purple top for the DNA testing. Of course, our main testing offered through the program is the comprehensive DMD gene analysis, which is performed by next generation sequencing. And this does detect both deletions, duplications, and smaller point mutations. And Dr. Hegde will be speaking more about NGS in a moment. We also offer targeted testing for known familial mutations. This is, of course, for carrier testing and for male SIBs of affected children. And we offer the neuromuscular panel, which I will discuss on the next slide. So new just this month, we are really excited to announce some of the changes that we've made to the Decode program, including a more streamlined approach to ordering testing directly through the PerkinElmer website. So we have actually eliminated the application process through PPMD, except for carrier testing and New York State testing. But we think eliminating the application should make the testing process even easier for clinicians. And if you are used to starting the process at the PPMD Decode website, you can still start there and you will be linked to the PerkinElmer site for ordering. Also new in 2020, we have added reflex to the neuromuscular panel for negative results. This is a 132 gene panel. So if you have a patient who tests negative, we will contact you about initiating this panel. And all of the details of the new ordering process through the PerkinElmer website will be discussed later in the webinar. Next slide. And before I turn this over to Dr. Hegde, I do want to emphasize that PPMD will continue to coordinate Decode Duchenne and offer genetic counseling support to clinicians, patients, and families. We have 2 board-certified genetic counselors at PPMD with extensive Duchenne and Becker experience, and we are always available to answer your questions or talk directly with your patients. And we've received a lot of positive feedback over the past several years about how helpful the free diagnostic testing and carrier testing is for both the clinicians and, of course, the families. So with that, I will turn it over to Dr. Hegde.
Thank you, Anne, and thank you all for joining today. As Anne mentioned, I'm going to talk about the science behind the program. We have come a long way in molecular testing, and it's really an exciting time in genetics because the technology has changed significantly, which has allowed us to develop methods which are cost-effective and more accurate and precise. So what you see on this slide is just a very high-level view of different genes and the mutation spectrum or the pathogenic variant spectrum of different genes. So on the left-hand side, you see genes such as CF and APC, where majority of the pathogenic events are small changes. And the Duchenne gene or the DMD gene is really unique in that aspect, where it's on the right-hand side, you can see that majority of the pathogenic events are deletions and duplications, followed by point mutations. Justin, can you just click on more? Thank you. What has happened since then also is that with the genome sequence being completed, many new genes have been identified. And these genes have been sort of added to different gene panels. And as just mentioned, the 132 gene panel for neuromuscular disorders. This contains all the new genes that have been identified, but we really do not know the spectrum or the variant spectrum of these genes, meaning what are the more common events in these genes? Is it deletion, duplication, or the smaller events? With the technology changing to NGS, we have been able to now move in that direction. So if we quickly look over the molecular methods that have been used for DMD analysis over the past 15, 20 years, my PhD thesis used the 32 exon multiplex PCR, and to where we are today and what we can do for the patients is really sort of a big milestone in terms of the technology and where we are. So what you see on this slide is the multiplex PCR. It was limited to 32. Some people started doing 79. MLPA is still quite widely used, especially in Europe. Sanger sequencing is used only for targeted events mostly. In between, in our later sort of between 2005 and 2010, the SCAPE method by Kevin Flanagan's lab was published, which is really single condition amplification internal primer method. Southern blotting for deletions and duplications, and then array CGH, which started getting used more commonly for detecting deletion duplication. But in all these methods, what you see is that either you detect deletion duplications or small changes or point mutations, and this is a two-tier test. You first do deletion duplications and then you reflex to sequencing or some other method. Which there are two things that can happen in doing this. One is you can lose the patient to follow up, that the second test does not get done, and we had published some data on that. And also that because these are targeted testing, by that I mean looking at the 79 exons and the flanking 50 base pairs of the intronic region, you're not detecting the full spectrum of the events in the DMD gene. And that's when really NGS really comes into play. And NGS as a method in clinical testing was used first in about 2010, and it has come a very long way in terms of its accuracy, sensitivity, and specificity. So with that, the method can actually detect the small events, deletions, and duplications. And by small events, I mean the nonsense pathogenic variants, small indels. And then if we can sequence the entire gene, then the deep intronic pathogenic variants as well. What we have also been able to do with the NGS method is validate it on whole blood, saliva, dried blood spot card, amniotic fluid, and CVS. I think one thing which really stands out here is the DBS card. We all know that we are making big strides towards getting DMD included in newborn screening. And the expectation is that if CKMM returns a positive result, that we can reflect to the molecular testing. And this is where the DBS card becomes very important, because the sample does not have to be collected again. Next slide. So I have practically used all the methods I described on the slide, as I mentioned, and really getting this NGS method validated was really very exciting. And the reason for that is the method by itself allowed us to sequence the entire 2.2 megabase of the DMD gene at a cost less than all the other methods that are described there, including the two-tier approach. So what you see on this slide is, you can imagine the blue bar being the entire DMD gene. And what you see is the Sanger amplicon or the MLPA, where the MLPA is specific probes spread out only in the exon region and a little bit into the intronic region. The Sanger amplicon or sequencing, and even some NGS-based methods, only sequence the exon and the flanking, the two blue arrows, which is about 50 bases. What we were able to do is to put probes or baits across the entire 2.2 megabase of the DMD gene. Now, obviously, the question that will come to your mind is, why sequence the entire 2.2 megabase of the gene? And I'm going to show you results which would justify why this is a really important thing to do and how it covers the entire pathogenic variant spectrum in the DMD gene. Next slide. So the first point I'm trying to sort of make from this, justify the entire sequencing of the gene, is the determination of accurate breakpoints. So we just heard Anne mention the different type of therapeutic intervention that are being developed for DMD, and one of them being exon skipping. There are CRISPR methods that are being developed. And what I've shown you on this slide is 4 different events: a single exon deletion, multi-exon deletion, duplication, single exon duplication, and multi-exon duplication. So let's look at the deletions first, and those are shown in red. And what you can see there in the figure A is that if you just focus on one line, which is the single exon deletions followed by the multi-exon deletions, what you can see is for the same event, the breakpoint can be different. So not every exon 44, 45, 51 or the multi-exon deletions have the exact same breakpoint. Why is this important? And I'm going to explain that with an example. A few years ago, I was sent a sample where the physician had said that the result given was Becker type event, but actually the child looked like a Duchenne progression. So we were sent a RNA sequencing sample where it showed that it was not actually that single exon which was deleted. The deletion extended into the intron, and which actually took out the adjacent exon as well. This is really important because this can affect clinical trials and outcome of the clinical trial. We all know that we always get that variation. One is the different endpoints we use for the clinical trials, but having this precise information is very helpful when selecting the individuals for the clinical trials. The same thing you can see in the blue bars for the duplication, where when you look at a single event, the breakpoints are different, so the bars are of different sizes. So this really shows you the power of doing the whole gene sequencing of the entire 2.2 megabase of the DMD gene. Next slide. So sort of spending a few minutes on the actual method we are using in our lab. As I mentioned, the CKMM assay is done from dried blood spot cards, and we can actually extract DNA from the DBS cards and do the NGS method as well. And from there, it goes right up to the whole blood sample. So we have designed a bait library using Agilent Sure Select technology, which actually covers the entire 2.2 megabase of the gene. We do not have any gaps in the bait, so that means we are able to cover the gene end to end. We sequence on a Illumina NovaSeq 6000 with 100 base pair and reads. Deletions and duplications and the sequence variants are assessed simultaneously on 2 different software programs. For deletions and duplications, we use the NX 4.3, And for sequence variants, it's our own proprietary sequence interpretation pipeline, which we call ODIN. And ODIN stands for Ordered Data Interpretation Network, which presents to the director the sequence variant and all the different databases for to enable clinical interpretation. We launched the clinical assay through our PerkinElmer Genomics lab after a thorough validation using different sample types. The manuscript for this validation and the results from our first about 700 samples are ready, and we are about to submit it hopefully on Friday to the journal Human Mutation. Next slide. So essentially what we are doing now is we do not need to do a two-tier assay. In one single experiment, we can detect deletions, duplications, and small events in the DMD gene, but we can also look at the intronic regions. And I'll talk of why that is important. So since the launch of the assay in 2017, there are about 275 samples, we have been received for diagnostic testing. And a diagnosis was established in 75% of the cases. As you can see, they are split for deletions, duplications, and sequence variants. Single exon deletions were in 8% and duplications in 1%. We also identified whole gene deletion. Again, this is a very important point here, and I'm going to show you some data on that of why this is really critical and how NGS actually facilitates this detection of the entire DMD gene when that event has occurred. Next slide. So we're going to go through some results and just to sort of display the clarity of the data and how the data is visualized by a clinical laboratory director. So what you see here on the left-hand side is a single exon 45 deletion in a male. So in a male with a single X, you expect that there is no hybridization or no target selection happening in the region which is deleted. So what you see here in this pink area is the exon 45 deletion. And the visualization of the data is same as a microarray. So we take the NGS data and we convert into a microarray type visualization format using this NX software. Now this is equally sensitive for a single exon deletion in a female as well. And you can see that in the female carrier with single exon 55 deletion, what you would expect is that a 50% break, and you can see that in the log ratio where you have one copy and one copy is deleted. Next slide. Again, the question would be, is this sensitive for duplications as well? And going down to the smallest possible duplication, what you see in this male here is a duplication of a single exon 2 and a female carrier with a duplication of exons 3 and 4. Now, again, you can see the slight difference in the display of the results, where in the male we can see the bar is a little higher, which is you're looking at a single X. In the female, you have one normal X, so the bar will be a little lower. Next slide. So now taking that and going on to the multi-exon deletions and duplications, what you see here is a duplication in a male of exons 3 to 29 and a female carrier with a duplication of exon 3 to 9. One thing which I should point out on this slide is if you see that the black sort of vertical bars and the density of the bars above, that really shows you the coverage of the gene, and you can see the uniformity of the coverage of the gene, and when the duplication is present, how those bars jump up. But the density is important here because, as I showed in the design slide, that with MLPA and other methods, you are just placing probes across certain regions and not covering the entire span of the DMD gene. Next slide. And this is a entire gene deletion. As I mentioned, we had 2 patients with the entire gene deletion of the DMD gene. Now, when such an event occurs, we do confirm it with another technology, and here we have used MLPA. But the one thing I want to point out on this particular deletion, and I don't know how close you can see it, but this deletion actually extended beyond the DMD gene. And when we talked to the physician mentioned to us that this child actually looks more syndromic, and there could be involvement of other genes, which we were able to confirm because we could show with our method that the deletion actually expanded beyond the DMD gene, and then they ordered a copy number genome which looks at the entire genome, and we were able to precisely map the breakpoints of this deletion which extended on either side of the DMD gene. Next slide. So now moving on from, we looked at deletions, we looked at the sensitivity for deletions, duplications, the breakpoint detection, the accuracy of the breakpoint detection, the differences in the breakpoints for the same event, and we also looked at the detection of the smaller events in the DMD gene. Now again, going back to the design and emphasizing on this fact that we are sequencing the entire 2.2 megabase of the gene, is we can now detect the intronic variants in the DMD gene which are reported to be pathogenic. So if you look at the Leiden database or the human gene mutation database, there are manuscripts and papers documented in these databases which have reported deep intronic changes which have been detected by RNA sequencing to be causative of DMD. So what we have been able to do now is we subscribe to all these databases, and we have tagged these variants in our ODIN database. And what do I mean by tagged? We have downloaded these positions, we have read these papers, we have curated these papers, and we have tagged, and we have actually written out a description. So what happens when the data comes into ODIN? If such a variant is present, it is automatically highlighted, picked up, and presented to the director. So what I'm showing here is a exon 31 plus 36947, so 36 kb into the intron, a G2A change, which has been previously shown to be pathogenic. And this would this particular event actually leads to skipping of the subsequent exon. Now, about 1 to 2% of the events in the DMD gene are deep intronic events, which today are not detected because by using these alternate technologies, which are only focused on exons and flanking introns. So here we now have a truly comprehensive assay which can detect the entire spectrum of events in the DMD gene. We've not shown today, but we have also been able to detect an inversion in the DMD gene. Now this required some really complex bioinformatic analysis, but the assay is capable of being extended to detecting those 0.1, 0.001 type events that could also occur. Next slide. So concluding this part is really, it's important to use an assay which has the potential to detect everything in a single assay. And just using this assay in the clinical laboratory and being able to give those results is very satisfying, because we know that if the deletion duplication is not present, the entire spectrum has been screened and not going to lose any individual to follow up, or they're not going to get a result or have this lengthy diagnostic odyssey. The clarity of the results is really, I hope I've shown you with the display of the deletions and duplications, that it is equally sensitive for males for diagnostic testing and females for carrier testing. And this is really a huge leap forward in the different methods and the traditional testing methods for DMD. The cost is significantly lower than any of the methods I have described. And it can, because we were able to validate it on a dried blood spot card, it can be used for confirmatory testing, molecular testing as a follow-up to newborn screening for DMD. So with that, I'll stop and hand it over to Justin.
Thank you, Dr. Hegde. So thank you for that great presentation, Ann and Dr. Hegde. So my role of the presentation is just to give kind of a quick update and overview of some of the things that we've changed to both streamline and get you all aware of kind of some of the changes that we've made to the Decode Duchenne program at PerkinElmer. So the first thing that we've worked to do is to streamline the ordering. So as opposed to previously when an application needed to be submitted to PPMD before testing occurred, you can now order testing directly from PerkinElmer with a few slight exclusions or exceptions there. We also have a new dedicated test requisition for the program, so there'll be no confusion of what you're ordering. The exact menu is listed. It's very simple and easy to follow, and we'll show you that in a little bit. We've also added a new Decode Duchenne kind of landing page or program page on the PerkinElmer Genomics website, which gives kind of a one-stop shop for folks to learn about the Decode Duchenne program, access the requisition form, order collection packs, get those sent to the office or the patient's home, and then all the details that you need to work through the program. So on the PerkinElmer Genomics website, the Decode Duchenne page is actually located under our sponsored programs with a few others that we have. On the right here, you can see an image. It's just a very basic landing page that really navigates the person to what the program is, the history of the program, and what they can accomplish through the program if they so choose. You'll see here, this is a close-up of that page. One of the main things that I have here are specified or specific ordering instructions that we've developed for the program. When you click on that, it brings you out to a separate page. It gives you all the information that you really need to place your order. It gives you contact information with PPMD if there's questions. It can give you access to the requisition form and can give you all the information that you may need. When you click there on the requisition form, you'll see that there is a brand new requisition form that is branded for a Decode Duchenne program, and it is specialized specifically for this program. This program has 4 tests listed: the DMD creatine kinase activity, the comprehensive DMD test for sequencing and del-dup, targeted testing and Sanger confirmation, and it's very easy to follow and very easy to order. There's also program-specific requirements that you can see here of how to collect the specimens, so there's no confusion as to what type of specimens can be submitted for each type of test. And again, all of this is very specific to the DMD Decode Duchenne program. The last thing that I wanted to run through quickly is the ability to request a kit. With requesting a kit, you can click on that link and it'll bring you out to a specialized form for the Decode Duchenne program. You can order the type of kit that you want, send it to yourself, as well as requisition forms directly to the office. The alternative option is to be able to send a kit to a patient. So we've built an online submission form, which you can navigate to through the website. Simply, all you need to do is upload a completed Decode Duchenne requisition form by dropping it into the tool here on the right. Select that you want to send the kit to the patient's home, put in the type of kit you want, the patient's information, and we'll take care of sending that kit directly to the patient's home for sample collection for DBS or saliva. They send that back in. We match that up with the requisition that's already in our system, and we proceed with testing. So those are some of the changes and updates that we've made to the program. And at this point, this brings a close to our formal presentation of today's webinar. So at this time, Ann, Dr. Hegde, and myself will take a few questions about Decode Duchenne program, excuse me. Please feel free to submit your questions in the chat or question box, and we'll work to address as many questions as possible. If your question is not answered today's webinar or you have some follow-up questions, you can always email us at genomics@perkinelmer.com. So if there's any questions, we'll give it a few minutes to fill in, and we'll go from there. Any closing comments, Ann?
No, I think everything was covered very well. So thank you, Justin, and thank you, Dr. Hegde. And certainly if anyone thinks of questions after the fact, they can email us at the email on the screen, decode@parentprojectmd.org.